It is my great pleasure to introduce Dr. Linda Thomashow, who is a research geneticist at the USDA Agriculture Research Service Root Disease and Biological Control Research Unit at Washington State University in Pullman, Washington. She is a fellow of the American Phytopathological Society and has published over 185 papers and reviews. She is a collaborator with the CommGen Authentic Research Experiences Grant and will describe some of her associated research for us today.
You have heard from Dr. Bengera and Dr. Weller about the take-all story. And today I'm going to tell you a little bit about other antibiotic-producing pseudomonads in dryland wheat. And my story is going to include the genes that these bacteria have and also something about their ecology. We know that we have large-scale ecology for Pseudomonas in the Pacific Northwest and specifically in Washington State. And our work has focused on the southeast corner of Washington State in an area that encompasses the Columbia Plateau. In 2008 and 2009, we began to sample over 80 commercial fields within a total area of over 22,000 square kilometers. And we were specifically looking for fluorescent Pseudomonas bacteria in the soils of agricultural fields. And we measured specifically fluoroglucinol-producing bacteria, but also bacteria that produce another antibiotic called phenazine antibiotics. And we used a technique called HPLC mass spectrometry to look for the antibiotics themselves in the soils from these fields. So the large-scale ecology of these bacteria was determined specifically by looking for the antibiotic biosynthesis genes that they contained. And we looked for DAPG-producing bacteria indicated by the blue-colored dots, and phenazine-producing bacteria indicated by the yellow dots. And the size of the dot tells you the relative amounts of bacteria that we could find on the roots of wheat colonized by the antibiotic producers themselves. If the blue dots contain the letter I, that indicates that the field was irrigated. And the most striking thing that you will notice about this map is that the blue dots are clustered mainly in the eastern part of Washington State, bordering on Idaho. When there are blue dots more to the west, in the center part of the state, those dots have eyes in them. The only fields in which we found DAPG producers in the west were irrigated fields. In contrast, in the west, the yellow dots were clustered, and those are phenazine-producing bacteria. So what are these phenazine-producing bacteria? Well, it turns out these were the first bacteria that we studied back in 1988. And in a landmark paper by Dr. David Weller and myself, we showed for the first time conclusively using genetic techniques that antibiotic-producing bacteria were responsible for the control of a root disease known as take-all. And in this study, we created mutants of a phenazine antibiotic-producing strain called 279, Pseudomonas fluorescens 279. Pseudomonas fluorescens 279 produces an orange compound, which is phenazine 1-carboxylic acid. And the colonies of bacteria producing this antibiotic have an orange color. And you can see in the middle of these petri dishes, this is the take-all fungus and there. And in the first plate, the bacteria are inhibiting the growth of the fungus, and so the fungus does not grow over near the colonies. In the second plate, there are some white colonies. These have been mutated no longer to produce the phenazine antibiotic, And the fungus grows over and overgrows those colonies with time. Phenazines are a really interesting class of compounds that have been studied. They were first noticed by scientists and microbiologists over 150 years ago, probably because the colonies that produce these compounds gave rise to very nice colors. And of course, they were eye-catching. And in the lower part of the slide, you see some of the range of colored colonies that produce phenazine antibiotics. In the bottom is a dark blue producing compound, a dark blue compound, and this is produced by a strain of Pseudomonas called Pseudomonas aeruginosa. This is a human pathogen. When this organism colonizes the lungs, usually of immunocompromised individuals, the disease is usually fatal because these bacteria most often are multi-drug resistant. But in the soil, phenazine-producing bacteria can be very beneficial. And they produce compounds. You see a range of the kinds of compounds that can be produced. All of them share this common tricyclic core structure. And then you can see from the structure that there are very many places on the structure where additional chemical derivatives can be added on. And the addition of those compounds leads to changes in the physical and chemical properties of the phenazines. And those changes also affect the target range or the antibiotic target range of the compounds themselves. So we became very interested in the synthesis of these compounds because it was a mystery since over 150 years ago how the compounds themselves were synthesized. And chemists had approached this problem through the use of radioactive isotopes, and they ran up against a wall with this technique in the early 1980s. And it wasn't until molecular genetics came on the field and the ability to clone the phenazine biosynthesis genes happened. And that was first done in my lab by a graduate student at the time named Dmitry Mavrodi. And after the first phenazine genes were cloned in strain 279, many other phenazine genes also became cloned from other bacterial species. And many Pseudomonas strains have phenazine genes. Occasionally, other bacteria, Brevibacterium, and some gram-positive bacteria, some strains of Streptomyces produce phenazines. But until this day, phenazines are unique to bacteria. These bacteria live not only in soils, but they live in the sea, they live in rivers. But most bacteria don't produce phenazine, so it's a rather special trait. And the pathway for the synthesis of this core molecule is very highly conserved. And specifically, the Fez-F gene is a unique gene to this pathway. So much the same as the Phil-D gene is a unique gene to the fluoroglucinol pathway, the Fez-F gene is a marker for the phenazine biosynthesis pathway. And we can track this gene in the environment using PCR, and we can monitor populations of phenazine producers in all sorts of natural environments. Also, from the sequence of the phenazine gene, it was possible for the first time to predict the protein structure. And we call this structure the kissing butterfly structure because the protein exists as a dimer. There's two copies. Each one looks like a butterfly. And so these are the kissing butterflies that actually carry out a key reaction in the phenazine biosynthesis pathway. Where did these genes come from? Well, it turns out that among the Pseudomonas, they're highly conserved, and they didn't come from anywhere. The Pseudomonas have their own. However, in certain other bacteria, like the Burkholderia species and Pectobacterium, they got the genes from the gram-positive, from the Streptomyces bacteria. So this is an example of gene exchange taking place in nature. And we think that among the Pseudomonas strains that produce phenazines, the compounds have a very unique role in the metabolism of the bacteria, whereas in the bacteria like Streptomyces and Pectobacterium and Burkholderia, these are defense genes related to capturing and owning a piece of turf in the environment. So by understanding the phylogeny or the family tree of the genes themselves, which can be predicted from the structure, we can also predict where the genes came from in the strains that have them. And this is a very, very good representation of which kinds of bacteria produce phenazines. Not all bacteria have this unique ability. Well, we used the sequences of the genes, and we tracked the abundance of bacteria containing these pathways in the environment. And I should point out that there are no natural phenazine producers that also produce fluoroglucinol, and there are no fluoroglucinol producers that also produce phenazines. So they are unique markers. And what we found as we surveyed all of the fields in the southeast corner of Washington State was that as annual precipitation increased, the number of phenazine producers declined. So only in the dry soils, closer to where the Cascades are in the desert in the central part of the state, did we find the phenazine producers. This correlated exactly with the map that I showed you with the colored dots. And as rainfall increased, as you went more to the eastern part of the state, more towards where Pullman is, then the number of fluoroglucinol or DAPG producers increased. And it was possible to do this big-scale ecology study because we knew the sequence of the genes that were unique to the various bacteria. We also could show by isolating phenazine 1-carboxylic acid directly from the soil that there were very significant concentrations of antibiotics produced on the roots of wheat grown in farmers' fields in this area. And as the number of bacteria in the field increased, so did the size of the amount of antibiotic that we could recover from those soils. We also measured the half-life. How long does that antibiotic persist in the soil? And we found that the antibiotic, on the whole, in most soils, was half gone in 3.4 days. So after 6 days, 3/4 of it would be gone. After 9 days, almost all of it would be gone. So this suggests that under field conditions, at least under the conditions that we were using in lab, that the antibiotic does not stay in the soil for a long time, and therefore the bacteria must be producing it over a significant period of time. Now, to reproduce these results under more dry land conditions, we've had to invent dry land conditions in the laboratory that we can study. And I don't have time to tell you about that work today, but it is not easy to develop dry land conditions under a laboratory environment. I can assure you of that. So I had another student in my laboratory named James Perico, and he isolated thousands and thousands of phenazine-producing isolates from the soils of southeastern Washington State. And he studied the diversity within that FES-F gene as a marker for the genetic diversity of these thousands of strains that he had isolated. And he could show, doing a family phylogeny of the phenazine genes, that they clustered into 4 major families. 2 of these families we named Pseudomonas aeridus and Pseudomonas cerealis because they were significantly different from any known Pseudomonas that was previously described. These are new species, unique as far as we know to dry land regions. Two other species groups we called P. orientalis and P. cinzantha because they had previously been described. Specifically interesting is Pseudomonas strain 279, the strain from 1988, fell into the cinzantha group. That was very interesting to us because cinzantha is one of the strains that previously was sequenced. So now we are very interested to understand the genomes of members of all of these major species groups, to understand what pathways may allow these particular bacteria to be so highly adapted to very dry ecological conditions. So we are sequencing representative isolates from all of these groups. I can tell you that we have draft sequence for Pseudomonas fluorescens strain 279. It's a typical Pseudomonas fluorescens strain. It has a rather large genome, over 6 megabases. The GC content is close to 60%. These are very high GC organisms. And so far, we have not had time yet to analyze the sequence to know what are the unique genes here. But as our sequence analysis has persisted, we see that much of the genome does resemble Pseudomonas cinzantha BG33R. And this is interesting because Pseudomonas cinzantha is in a totally different group within the Pseudomonas family compared to Pseudomonas brassicacearum, which is the new name for L51, strain L51, the strain that you are sequencing. So these isolates, these strains, are very different from each other. And that's totally consistent with the fact that Q8R1 and brassicacearum like to live in soils that are reasonably moist, whereas strain 279 and potentially other cinzantha strains are more highly adapted to dry soils. So this tells us something about the ecology of these strains and where we might expect to find them in the future. What about the practical importance of these phenazine producers? I told you already that phenazine producers are inhibitory to Gumanomyces graminis, the take-all pathogen. But in fact, they don't live in the same soils where the take-all pathogen is very important. They prefer drier soils, and the take-all pathogen prefers wetter soils. In fact, under the non-irrigated conditions where these phenazine-producing strains occur, Rhizoctonia solani and Rhizoctonia oryzae are the main kinds of pathogens that we are concerned with. And these pathogens cause important root rots that Dr. Bengera mentioned earlier, and they cause these bare patches in the field that you can see in the slide. Well, it turns out that these phenazine-producing bacteria are very inhibitory to Rhizoctonia oryzae, which looks like a little animal skin here in the middle of this petri dish. You can see the zone of inhibition very clearly in this plate. And the control of the disease is common to members of all 4 groups of Pseudomonas in this cluster of strains that is inhibitory under dry land conditions. So we were very interested in understanding more about the impact of water availability on the composition of the microbial community in the wheat rhizosphere under irrigated or dry soil conditions. So we received a grant in 2011 to understand the community structure of the bacteria on the roots of wheat growing either under very dry conditions in Lind, Washington, or in the right next field under irrigated conditions. So we had set up dry and irrigated fields adjacent to each other. And this slide shows Dr. Weller in the field establishing the soil moisture measuring equipment at the time that the field is seeded in the spring, and then the wheat early in the growing season when the rows are still readily apparent, and later in the growing season when the whole field appears to be full of wheat. And we are tracking the dynamics and activity of the microbial communities over a growing season in these plots, and then we are doing community analysis to understand what microbes are present under both of those conditions. And here the slide is showing that the phenazine-producing bacteria decline in population size over the growing season, and the amounts of phenazine that we can recover from those fields also declines as the populations decline. Moreover, the irrigated plots lose their populations of phenazine producers faster than the dryland fields do. We also see that as the sampling date goes through the season, the relative numbers of phenazine producers on the plants increases with increasing time. So those populations are enriched as the soil becomes drier. And this is totally consistent with their preference for dry soils. So we're also doing soil DNA extractions from these fields. We extract the phenazines. We also extract soil DNA. There are 60 soil DNA extractions per sampling, 3 soil DNA sampling times per year. So that's 180 DNA isolations from soil every year. And then PCR with 16S and phenazine gene primers out of those enriched samples, and prepare the samples for metagenome sequencing also. And this work is in progress. In fact, those samples were sent off the last week of October to Texas A&M University for sequencing. And I hope that by March of 2015, we will have the first data that shows something about the microbial composition of the soil under dry land and irrigated conditions in soils colonized by the phenazine producers. Specifically, we would like to know if the phenazine producers have a role in shaping the structure of the microbial communities under those two conditions, whether irrigated or whether dry land. And I thank you very much for your attention.